mapping generates a BUM or a SUM file. So no longer FASTQ files, but BUM and SUM files. Some mappers produce a BUM or a SUM file where the reads are in the same order as they were in the FASTQ file. So the order of the clusters on the flow cell, basically. Now, for many downstream applications, this is not the best choice. So what you're going to do is, for instance, visualize the results of the mapping, and you're going to visualize along the genome sequence. So it means that you are going to visualize the reads from chromosome 1 first, and then the reads that align to chromosome 2, and so on. So in that case, it's much more helpful if the reads are sorted according to the location that they map to. The same is true for counting, for instance. You're going to count in the next steps, and typically also there you're going to count along the genome sequence. You're going to count how many reads map to certain genes. You're going to count how many reads map to certain genomic locations. So also there, it's much more useful if the reads are sorted according to the genomic location that they align to. First the reads from chromosome 1, then the reads from chromosome 2, and so on. So there are tools that you can use for sorting BUM files. So if the mapper hasn't sorted your BUM file yet, then you can do it using a tool that can sort a SUM or a BUM file. You need to index your BUM files. So these are very huge files because they contain all the information from the FASTQ file, and on top of that they contain the information from the mapping. So they are very huge, and it means that they will be very hard to access and to search through. So just like we have done with the reference genome, we have built an index to make it easier to access and to search in. We're going to do exactly the same thing with our BUM files. We're going to index them, and here in this case the index will be stored in a separate file that has exactly the same name as the BUM file, but it has the extension .by instead of .bum. For DNA-seq applications, you also have to mark or remove the duplicates. So in DNA-seq, duplicates are thought to arise from technical issues, mainly by PCR. So they are not biological copies, and as a result, they will distort the counts. So you have to remove them before you count, and any application involves a step where counting is done. So in ChIP-seq, you will count the reads that align to each genomic location, and you will search for genomic regions with many reads in the ChIP sample, the so-called peaks. Tools for finding peaks know that the duplicates are an issue and they will ignore them. But the downstream tools for visualization of the mapping results and the peaks cannot handle the duplicates. So you still need to remove them yourself, or at least mark them. And marking means that you're going to indicate in the BUM file which reads are copies. Tools that identify variants. How many times a variation occurs. So here also you want to remove the duplicates. So the variant callers can, however, not handle the duplicates on their own, so you have to remove them or mark them yourself. For RNA-seq, most duplicates are believed to be biological copies, so you do not want to remove them. So, but these are typically the 3 steps that you do to process your BUM and your SUM files. So there are many tools that allow you to do so. The most used tools for doing this are called SUM tools and PICAR. You will also check the quality of the mapping. Just like you checked the quality of the raw reads, the quality of the sequencing basically, you're also going to check the quality of the mapping. And the tool that is used the most for doing that is called QualiMap. And it's very similar to FastQC. And then the nicest part of the whole workflow, you're finally going to visualize something. So you're going to visualize the results of the mapping. And for that, we're going to use a tool called IGV. So when you sort and index your BUM and your SUM files, what is generated? Well, still a BUM or SUM file, typically a BUM file in this case because they are more compact. But the order of the reads in the BUM file has changed. So it's a sorted BUM file, and when you index your BUM file, a separate file is generated, and that separate file is called a BY file. And BY stands for BUM index. So the two tools for doing these kind of manipulations on your BUM and your SUM files are called Picard and SUM tools. So it's not one tool, they're basically both a set of tools, and they will have tools for sorting and indexing BUM and SUM files. So for Picard, this is called sort SUM. In some tools, this tool is called sort. You will have tools for just doing the indexing on a file, a BUM file that is already sorted. And Picard also has a tool for marking duplicates. So in RNA-seq data, you will have many biological duplicates, but of course there also will be technical duplicates. There's not really a way to avoid that. But because you cannot make the distinction, people say, oh, we're going to play it safe and we're not going to remove them. We're going to assume that they're all biological. That's the best thing that you can do here. Now for DNA-seq reads, you don't expect to see duplicates. So there people do remove them because you don't expect to see biological duplicates. So when you see duplicates, they're probably technical duplicates.